Sir is a completed MPharm in pharmaceutics and PhD from Kelly University. Sir has got experience of 10 years, out of which 5 years is in industry as an research and development officer and 5 years in academics. Sir received AICTE scholarship for his post-graduation from the year 2008 to 2010. Sir has got various publications in national and international journals. Sir has presented various papers in national and international conferences. Sir is also a member of International Society of Ethnopharmacology, Indian chapter. His areas of interest is self-emulsifying drug delivery system. So today, Sir is going to talk on self-emulsifying tablet, promising drug delivery system. With this brief introduction, I welcome you to this session, Mukund sir.
Yeah, thank you, ma'am.
Over to you, sir. Thank you.
Okay. Okay.
Okay. Please share your presentation, sir.
Yeah, I'm sharing.
Okay, sir.
Can you see my screen, madam?
Now you share your screen, sir.
Okay.
Sir, down there is a toolbar, no? There you have to click
Down.
share the screen.
Yeah, yeah, yeah. So are you getting my screen?
No, sir. We can see you, sir, but
Okay.
presentation we cannot see.
Wait, wait. So I have to go through Drive or somewhere. So there is no option I am getting that share screen.
Just 1 second, sir. We will help you out.
Hello, sir. There is an option share screen in the Zoom app.
Okay.
Presentation mode. Yes, sir. Okay.
Yeah.
We can-
Yeah, we can see the screen now, sir. You can start the presentation.
Okay. Can I start, ma'am?
Yes, sir.
Yes, ma'am. Can I start now?
Yes, sir, you can start now.
Yeah. Yeah, very good morning, one and all. So first of all, I would like to thank Kelly Society's College of Pharmacy, which is my family, for giving me an opportunity to share my some small research ideas or views regarding the topic self-emulsifying drug delivery system. In fact, which I'm going to focus more on self-emulsifying, the developmental aspect of self-emulsifying drug delivery system as a part of self-emulsifying tablets. So, thank you. So, I would like to move ahead. So, these are the contents of my presentation. So first of all, we'll see the uh, introductory things. Then we'll see the formulation components or formulation aspects of self-emulsifying drug delivery system, and how actually the mechanism of self-emulsification or self-emulsification occurs. So in developmental part of this sets, then the development of self-emulsifying tablets, then evaluation aspect. So we'll cover few evaluation parameters as a time constant is there, then summary and conclusion from the research work. So actually today I'm going to talk about the topic which is SADS. So we'll see what is the exactly the self emulsifying drug delivery system is. So it is defined as an isotropic mixture of oils, surfactants, co-surfactants, solvents of natural or synthetic origin, which may be in solid or liquid form. So this is actually the uniform mixture in simple language of oils, surfactant and co-surfactant, in which the drug is available in the form of solution state or completely mixed state with all these three components. These are actually anhydrous liquid mixtures called as pre-concentrates. So we'll see how what are the actual components of this seeds. So formulation components of the self-emulsifying drug delivery system includes active pharmaceutical ingredients or drugs, then oils. This oil may be synthetic or from synthetic origin or natural origin, you can select. Then surfactants and co-solvents. These are actually basically surfactants only. You can call co-surfactants also. So these are the four basic components of self-emulsifying drug delivery system. Then next, so we'll see the mechanism of self-emulsification. So how this self-emulsification occurs, or why there is name self-emulsification. As the name indicates, self-emulsification. So actually it is the based on this occurs on the basis of fact that how the entropy of the system changes. The free energy of the emulsion formation is based on energy required to form new interfaces formed between two phases. So actually, this is the isotropic mixture. When this mixture comes in contact with the solvent system, suitable solvent system, there is a emulsification process occurs, and this energy required for the emulsification is very low. So that is the reason this emulsification occurs automatically, we can say. So this surface free energy can be calculated by the given formula, where the delta G is a free energy of the process, which is very low, or you can say minimum. Then n is number of droplets formed in the emulsion and radius of that droplet. Then sigma is interfacial energy of that system. So this mechanism of self emulsification occurs because of it requires very less energy or low energy. So this is all about the how the self emulsification process occurs. You can calculate the delta G or the surface free energy of the system. Then you can predict the how fast or how slow the emulsification is going to occur. Then next slide. So coming to the point, so why this self-emulsifying drug delivery system or self-emulsifying tablet is attracting attention of the researcher nowadays. So first of all, this system are having this system is having certain advantages. So first is drug, one can protect the drug effectively by designing this type of system. Then it is possible to deliver the drug at targeted site. So if you compose the proper formulation, final formulation after the development of the liquid seeds, then this type of drug delivery system is having high drug loading ability. So if you compare the another one technique, that is liquid solid drug delivery system, so there is certain limitation of carrier. Each and every carrier is having particular characteristic feature to acquire or to get the drug loaded on it. So here there is no limit. There is a basically it depends on the type of surfactants and co-surfactants or oil system that you are going to select. So actually the basis for selection of this oil and surfactant and co-surfactant system is the solubility. So that part we are going to discuss in next slides. Then stabilize it this system stabilizes drug thermodynamically. So this is another advantage. Then fine oil droplets minimize the irritation of gut wall. So because of very minute or fine mist of this droplets of emulsion will minimize the irritation of gastrointestinal mucosa. If you get the droplet size which is having higher size or larger droplets, then there is a chance of GI mucosal irritation. So if you prepare these things at a minute level, or if there is a very fine droplet size, this can minimize the irritation of gut wall. Then there is one of the greatest advantage of this technique. Nowadays, if you look at the industry or research and development department of the industries, so many project needs to have the skill ability, or there must be large scale production ability. Any technique or anything which you are going to develop at research level or in a laboratory level or at small scale, that must be having industrial application. So this is one of the technique which is attracting the attention of the researcher or formulation scientist because of its unique advantage, that is it has industrial application. In fact, the scale up can be possible. Suppose if you are uh, producing or designing or developing particular formulation at laboratory level at small scale, you can go for the large scale production of it because of it has industrial application. This is one of the unique advantage and one of the unique reason that the formulation scientists or industries are getting attention for this technique. So there is another one reason that why we have to go ahead to the tablet than the liquid seeds. So while designing or developing the liquid seeds formulation, this liquid seeds formulation can be finally formed or formally finally packed into the soft gelatin capsules. Actually, these things are very critical. There are certain reports and studies which are they have reported that the amount of active API which is available in the liquid cells formulation, it is going to deposit into the gelatin capsule shell, and that amount of drug is not able to release during the its after its administration, or if it is getting in contact with the gastrointestinal fluid. So deposition of this API in the gelatin shell is is leading to the decrease in the drug content or the variation in the drug content than the seeds actual seeds formulation. So this is one of the drawback, that one cannot stop at the developing only liquid self-emulsifying drug delivery system. After verification whether your drug is getting deposited in gelatin shell or wall or that gelatin shell cover, you have to decide whether you have to go or you have to mix that liquid isotropic mixture with the suitable carrier, and you can take this liquid isotropic mixture mixed with the suitable carrier for the further compression, or you can move for the hard gelatin capsule filling. So based on that, the tablets are suitable measure that one can compress these tablets in a simple way or in as per routine procedures. So these are the reason why one should go for self emulsifying tablets. Then this is the schematic presentation that how one can go for it. So first of all, you will find the drug solution. That drug solution you have to prepare into the oil phase, and you have to mix up with that drug with the oil properly till the homogeneous dispersion or transparent dispersion is formed. Then you have to mix remaining co-surfactants and oil components further, so that there will be complete blend of that mixture. So here only you can find the exact composition of drug, oil, surfactants and co-surfactants. So surfactants and co-surfactants are used to ensure the complete dispersion or dissolution of the drug into the oil phase. So you have to use surfactants as well as co-surfactant or co-solvents for this process. So you can find the how exactly the surfactants and co-surfactants orient themselves in the system. So at the center you will find the drug is accommodated in the system, and you will find the actual orientation of surfactant, that is hydrophilic head and hydrophobic tail, in the oily region, where if you disperse or if you add this drop into the aqueous phase, you will find the separation or emulsification of this process. So this is the graphical view that how you can go for, or how exactly the process is carried out to prepare the self emulsifying drug delivery system. Then, so just for as a case study, we have selected one of the API, that is orlistat, as a model drug to prepare the this self-emulsifying drug delivery system or tablet of this orlistat. So orlistat is model drug used for study, which is used in treatment of obesity. So it is gastric and pancreatic lipase inhibitor, which is obtained from the Streptomyces toxotricini. So we'll just for a to study or to have a look on a particular case how this drug candidate is studied before selection of this drug candidate to the this drug delivery system. Then we'll see what are the basic problems associated with this API. The therapeutic point of view, if you look at this API, which is having very poor, which is poorly soluble, and because of which there is poor lipase inhibition. The studies reported that only less than 30% of this 30% of lipase inhibition occurs, then actually multiple dosing frequency is required, then per day cost of treatment is more. So by considering all these parameters, that one has to decide or one has to think about the newer drug delivery system. Then another parameters which we have considered during the selection of this model drug or API is basically this drug is very vaccine nature. So because of this waxy component or waxy nature of drug, one cannot directly combine or directly prepare the tablets of it. So you have to think differently if you are thinking to compress this API as per routine compression procedures. So this is one of the drawback or limitation. Then another problem of this drug is associated with, that is very low melting point, that is 44 degrees Celsius is the melting point of this technique, this drug. So because of this drug is very suitable or right candidate for the self emulsifying drug delivery system, because this drug delivery system does not involve any kind of heat procedures or heating or use of temperatures. So this technique can ensure the purity of API till the dosage form development or up to the final stage of dosage form. So this technique is devoid of heat, or this does not involve any kind of heating procedures. So the low melting point drugs are the best candidate for self-emulsifying drug delivery system. Then conventional oral capsule having repeated dosing frequency and only can inhibit 30% of lipase. So just which we have discussed already in therapeutic. So normal, the marketed formulation, which is conventional, which is available in market, it is having larger dosing frequency than the expected, because it only inhibits the 30% of lipase. So these are the certain problems associated with the API. We'll see the next slide. So the aim of uh, the research work was to prepare and evaluate self-emulsifying tablet of orlistat to enhance the solubility. So this was the basic aim which was considered or which was targeted during the study. The We'll see the schematic presentation or developmental stages of self-emulsifying drug delivery system. At the beginning, we have to screen or we have to conduct the certain preliminary trials to determine or select composition of oil, surfactant and co-surfactant. So this task is effectively carried out with the help of construction of ternary phase diagram. The basic principle or basic purpose of construction of this ternary phase diagram is to find out the best suitable composition of all three uh, ingredients or all three formulation components. So this is the basic aim of construction of ternary phase diagram. So after and another thing, this diagram will help you to identify the correct micro emulsion region, or at which point or at which composition exact micro emulsion is formed. So then formulation and optimization studies for the self-emulsifying drug delivery system by 3 raised to 2 factorial design. Okay, so two parameters were considered for this, considered as an independent variable, and two parameters were considered at dependent variable. So based on that, this 3 raised to 2 full factorial design is used to screen the exactly optimize or to finalize the one single formulation from the different 9 batches. Then evaluation of these sets was carried out for the parameters like global size measurement, then size distribution, then emulsification time, then transmission electron microscopy or TEM. Then this liquid sets formed will be further subjected for the freeze drying of the sets. Then evaluation of these freeze dried sets for FTR, DSC and XRD and SEM studies. Then finally this freeze dried cake will be is mixed with the suitable diluent mixture and compressed by using this compression machine. So this compressed compacts of self-emulsifying tablet were evaluated for the in vitro drug release studies, as well as they are evaluated for the physical properties. So we'll see the things. These are the actually different chemicals or drugs from which we have procured or supplied as gift samples. Here capriol PGMCM was supplied from the Gattefossé, France, which was used in this study. Then we'll see the different methodology or techniques which were used for the analyzing the actual drug components, formulation components or excipients or API. So these were the techniques which were used during the study. So this is the design layout for the self-emulsifying drug delivery system. This is the 3 raised to 2 full factorial design, that is two parameters at three different levels. That is, first is quantity of x1 is quantity of castor oil, then x2 is ratio of surfactant to co-surfactant. So these two independent factors were kept at three different levels, that is -1, 0 and +1, and the composition of this system is changed accordingly. and 9 batches were designed for the study. So based on the composition, there are different outcomes which we are going to discuss in next slides. So this is actually regarding the pharmacodynamic evaluation of this final tablet in the rats. This grouping of animals and the parameters or treatments are shown in the table. Then we'll see the results and discussion. So as a part of pre-formulation study, we have studied for the lambda max of this drug, and as per the procedures mentioned, we have prepared the stock solution, and accordingly we have analyzed the things on UV. So this is the part of pre-formulation study. So in the pre-formulation study, we have also studied the actual peaks as per the chemical structure of the drug compound, whether they are going to retain or not. So those things were ensured or confirmed by the FTIR spectra. After analysis, we found that all peaks were retained, functional groups were retained after the FTIR study. These are the actually pre-formulation procedures. Then next is melting point. So melting point of the this drug is determined by using the DSC, that is differential scanning calorimeter. So it is observed that 42.77 degrees Celsius, which is near to the reported value, that is 42 to 44 degrees Celsius. So it has shown the endotherm at 42 degrees Celsius. Then the solubility determination. This is one of the basic and first step of development of self-emulsifying drug delivery system, one can say. So the solubility determination of this drug is carried out by taking the different vials or with the 10 ml capacity. In that, the selected vehicle is taken with the 50% of the portion, that is up to 5 ml, and the amount of drug is added till there is a no precipitate or no turbidity is observed. So meanwhile, these vials were shaken for 72 hours in the orbital shaker at the 37 degrees Celsius temperature. Then after the precipitation or turbidity, these vials were centrifuged at 8000 rpm and the supernatant were collected, where we have to determine the amount of drug which is dissolved or which is mixed in the particular oil, surfactant or co-surfactant. So this was the basic task or basic step that one should go for the solubility determination. So after doing that exercise, we found we have screened different oils, that is olive oil, sesame oil, castor oil, cottonseed oil, soya oil, corn oil and peanut oil. So this, after screening these oils, we found there is a better solubility in the castor oil. Then in case of the surfactant and co-surfactant, we found that the capriol PGMC, this is one of the surfactant which is obtained from Gattefossé, this drug has shown more than 800 milligram, near to the 900 milligram solubility per ml of this co-surfactant. So this was the biggest achievement, that the drug has shown much more solubility up to 900 milligram, which leads to, or this decides or this indicates that one should select this capriol PGMC as one of the surfactant or formulation component for the study. So here we have selected castor oil, capriol PGMC and this twin 80 for the further procedures. So we'll see the development of UV spectroscopic matter. So the things which we have studied in the initial to check the lambda max of the drug product. So we have confirmed or calibrated that method whether there is a reproducibility or not. So this is the data. So next is drug excipient compatibility study by differential scanning calorimeter. So by doing DSC study, we just checked whether drug is suitable or compatible with the excipients or not. So it has shown the endotherm at same uh, reading, that is 42.77 degrees Celsius. So hence it concludes that there is no any physical or chemical interaction of drug with the excipients. Then we have studied also FTIR, carried out the FTIR analysis of drug as well as excipients. From this study also, we have not found any chemical interaction of drug with the excipients. Then coming to the point, that is formulation and optimization of seeds. In the methodology, we have just discussed the actual formulation components or the design as per the 3:2 full factorial design. So at the beginning, we have screened the different oil surfactants and co-surfactants. From that, we have selected capriol, PGMC, castor oil, and twin 80, these three components. So the basic intention to construct the ternary phase diagram was to find out the microemulsifying region. So we have identified the things which are green in color in the ternary phase diagram indicates the microemulsifying region. So that composition decides, actually you can carry out the trials by varying the composition of these three components within that region. So this microemulsifying region gives an idea that there in that region only you can try furthermore batches to have the smaller size of seeds or droplets. So this is actual the composition design. Then characterization of seeds. So seeds were characterized after designing or developing this composition. The seeds were characterized for the global size, emulsification time, then polydispersity index, then optimized optical density of the isotropic mixture after emulsification. So 9 batches were prepared. So from the 9 batches, the batch which is having the composition, that is SO5, that is castor oil 20 parts and 80 parts of twin 80 in that, 30 parts of twin 80 and 50 parts of caprol PG. So that batch was found to be acceptable based on the optimization study. The optimization criteria was minimum global size and minimum emulsification time. So SO5 batch showed the minimum global size, that is 96 nanometer, and emulsification time in second is 26 second than other batches, whereas PDI and optical density was also found to be minimum or acceptable. So on the basis of the global size and emulsification time, we have optimized with the help of software, that is Strat Graphics. We have optimized this batch by setting the parameter or acceptance criteria or optimization criteria, that minimum global size and minimum emulsification time. So on that basis, we have optimized this batch for the global size. Then from the 3D response surface plots, we have also seen there is some effect or there is some impact of the selected components, formulation components, on the parameters or dependent variable, that is global size and emulsification time. Here we have got, as the quantity of oil increases, there is increase in the emulsification time as well as global size. So this quantity of oil is having showing the impact, that is positive impact on this size and emulsification time. So we have considered during this optimization, you can view the impact of your independent variable on dependent variable by plotting the 3D response surface plots. So in next slide, this finalized or optimized sets were subjected for freeze drying. By keeping the conditions, different conditions, we have freeze dried that optimized sets. Here 5% of lactose were used as a cryoprotectant to have the final cake in a proper format or in a proper way. So this was the assembly, and we have gone for the freeze drying. Then this further freeze dried product as well as liquid sets were compared whether both this uh, freeze dried product after tableting were considered for whether they are going to produce global size and same zeta potential, or are they going to form the stable emulsion, or how the emulsification occurs. So we have gone through it and we have checked for global size. So there is no, the significant change was observed after tablet formulation, or this dried cake was converted into the SAID's tablet. And after this reconstitution of this tablet, we have compared global size. We have not found any significant effect, as well as we have not observed any change in the zeta potential. So this indicates that after converting this freeze dried cake into the tablet, we have understood that, and we can conclude that there is no any problem, or there is no any change in the global size. Then we'll see the scanning electron microscopy of this drug product. So you, in first picture, you can find the surface topography of orlistat. Here in freeze dried formulation, you can find there is a smooth texture than the original pure drug, which indicates that there is a complete amorphization, or there is a complete solubilization of this drug into the actual mixture or system, where you will find there is rough topography of the pure orlistat than the freeze dried shades. So this is transmission electron microscopic pictures. Here, these pictures or things were taken at a there is a bar which is showing the 500 nanometer scale. So the results which were obtained from the nanotrack estimation for the global size, these are in collaboration with the transmission in electron microscopic readings, that it shows near about 100 nanometer size of the shades. Then this is the X-ray diffraction study of pure drug as well as freeze-dried cake or final tablet. So here you will find there is a complete solubilization or amorphization of the drug component after the formulation. So there is reduced intensity of peak is observed in the X-ray diffraction study. So this is the indication that drug is at solubilized state. Then drug excipient compactility were studied in the formulation by performing the analysis of orlistat FTIR analysis. So the basic functional peaks were retained at the values which were indicated in the orlistat. So this is the FTIR spectra. Then DSA analysis confirms the same thing, that it showed the endotherm. Actually, it shifts little bit, but it shows the exact near to the values. Then actually this freeze dried formulation, how this is tableted. So actually this self-emulsifying tablet was prepared by drying the liquid seeds by freeze drying method, and this freeze dried cake was mixed with the different components, that is diluents, that is Avicel pH 102, Aerosil 200, and sodium starch glycolate, and Flasdone F29. So these were components which were used for the study, and finally tablets were prepared. Then next, the in vitro drug release study of this formulation was carried out by using the dissolution media containing actually 3% sodium lauryl sulfate with 0.5% of sodium chloride is recommended, but we have screened all these things at 0% SLS, 1% is there for the 3% SLS. So we have just checked with the conventional capsule or marketed formulation which is available. So for 3% of SLS and developed a self-emulsifying tablet, as you can see in the figure B, SEOT, that is self-emulsifying orlistat tablet, that is compared for the dissolution with the conventional capsule or marketed formulation. So here we can find the difference between the initial stages, and for this drug to inhibit lipase, there must be sufficient amount of drug to inhibit the lipase. This lipase inhibition is time dependent. So if there is a no release or less release at the beginning to inhibit the enzyme, so there will be remarkable effect at the end. So this marketed formulation showed the less release or percentage drug release was observed to be less at the beginning time point, that is 10 minutes, 20 minutes. as compared to the self-amylspine orlistat tablet. So this was the conclusion. Then in vitro enzyme study, inhibition study was also carried out. So after the in vitro dissolution study, so this percentage lipase inhibition showed by this self-amylspine tablet was found to be more as compared to the marketed one. So this could be because of this initial less release or less release at the initial time point, that is 10 minute and 20 minute, as compared to the developed self-amylspine tablets. Then in vivo anti-obesity study was carried out in the rats. So from this study, you can find out there is a significant difference in the tablet, that is body weight was uh, decreased in if you compare the SAID's developed formulation as compared to the diseased one. Then from this, actually, we can summarize or conclude the early state was characterized for its solubility, melting point, and absorption maxima. The solubility of orlistat in natural oils, surfactants, and co-surfactants was studied. The purity of orlistat was confirmed by the FTR and DSC studies. The UV spectroscopic method for the orlistat has been developed, which is simple and suitable. The compatibility of orlistat with excipients was verified by DSC and FTIR studies, and which was found to be compatible. Then SAIDS was successfully formulated, optimized, and evaluated for different parameters. FTIR, FTIR and different DSC studies and in vitro enzyme inhibition studies. From the evaluation, it can be concluded that optimized SAIDS formulation showed maximum drug release than the marketed one. So optimized formulation was also compared with the another formulation, and which showed highest rate and greater extent of lipase inhibition than the marketed formulation, which supports increased drug release and dissolution property of poorly soluble orlistat. So this was the conclusion from the study. So thank you. If you have any question, you can ask.
Dear delegates, if you have any questions, you can ask. Also, you can put your questions in the text chat box.
Hello, Mukund sir, Dr. Mukund sir.
Yes, sir.
How much castor oil is there in one tablet?
Actually, quantity was very less. I think so. 15 to 16 micro ml, sir.
Micro ml. Okay.
Yeah.
End product is stable at room temperature of your freeze dried?
No, sir. Actually, there is another work is required to develop the suitable packaging system for this type of formulation, because this constitutes oily mixture.
Okay.
So we have to protect it in after loading onto the suitable carrier. We have to pack them in the proper packaging system. So that will be the another area of research for this type of formulation.
But you have not taken care in the formulation of the any of the antioxidant this one, because when you store these oils, especially castor oil, for prolonged period, it may go for the certain modification or peroxide effect might be there. But why you have not added any preservatives or antioxidants?
Yeah. That stability data, which I have not focused. But during the 3 months of accelerated stability study, we have not found any symbol of that degradation and all.
Because this contain, because castor oil is a resonolic acid, it is a double bond. And
Yes.
It is very prone for the oxidation.
Yes, sir. But during the 3 months of stability study, we have not found any that type of effect. So could be if we go for the real-time study or testings, that could the things which you are mentioning, that must be further studied.
But when you are converting that whatever the freeze dried product to the this one tablet,
Yeah.
I think you have to consider the all the loading factor, all those things of the excipient with or the
Yeah,
sorbate.
that is actually, sir. Loading of these things is not necessary to study. You can directly mix up, like liquid solid drug delivery system. You have to think on that, and there is a limitation of each and every diluent or carrier. That there is, it will not take more that API on the carrier. So this, after doing this, you can directly mix the things up to where you are going to get the proper flow or micromeretic properties of that cake. So directly you can mix with the this Avicel pH 102, and you just mix manually, and just you try to go for its evaluation, like flow properties, angle of repose, and CI HR, and on that basis you can go for the tableting. So there will not be any question of this.
Okay, what is the dose of orlistat?
60 milligrams, sir. 60 as well as 120 milligram.
What was the cut weight of the tablet?
So final weight was near about, sir, 500 to 400. That is actually the drawback, you can say, which I have not highlighted. But you have to reduce the minimum weight by studying different carriers. We have also studied the along with Avicel pH 102. We have studied fusicalin and nesulin. But the problem, the tablet weight was 160, 120 milligram with the nesulin as well as fusicalin. But the problem was the friability issues. So that things which we have not highlighted.
Okay, thank you.
Yes.
We have a few questions in the chat box. We will read them, sir.
Yeah.
There is a question from Vijay Pambulkar. It says, is this formulation planned for commercialization?
Yes, definitely. This is the actually one of the advantage of this technique. If you try to search in the market, there are many formulations which are available in the form of soft gelatin capsules. That is, liquid cells are packed in the soft gelatin capsules. Up to that extent, this formulation are commercialized or already. But to overcome the drawback of the deposition of API into the gelatin shell wall, we are exploring this kind of work to be taken at the scale up or to be taken at the industrial level. So definitely this technique can be having a scale up potential and can be commercialized, provided that there is a need to have a proper development of proper or suitable packaging system for this formulation.
Okay, sir. There is one more question from Anand Bhandarkar. That is, what are the criteria of API to be formulated as self-emulsifying drug delivery system?
So basic purpose of this technique is to achieve the targets of solubility or dissolution. If you are considering the API which is having certain problems, or the dose is one of the factor, but as per my views, there is no limitation that you can try to screen maximum number of surfactants as well as co-surfactant. Then you can also overcome the limitation of the dose. You can incorporate maximum dose in the system, because if you having greater solubility of the API, the maximum solubility of API can be screened by checking the n number of oils and surfactants, and from that you are going to select the highest solubilizing agent or the component which shows the highest solubilization for your API. So by doing this, dose is not a criteria, but you can select the API those which are having very low melting point. This is one of the criteria which I suggest based on my views.
So there is one more question from Sonia Naik. It is so just wanted to know whether there is any specific requirements while selecting the API for SEDDS.
Uh, as such, you have to select if your intention must be the solubility issue or dissolution enhancement, as I said earlier. But if you are considering some technical problem of the API, just now which I have mentioned, that is melting point and all, so you can go for it. The low melting point API you can wonderfully formulate into the form of sets. That is the achievement, or that is the benefit by if you go for this kind of techniques.
Yeah. Any other delegates wants to ask any question?
Hello.
If there are no questions,
Hello, am I audible?
yes, sir. Yes, sir. Please go ahead, sir.
Hello.
Yes, sir. We can
Yes.
hear.
Okay. So very good and informative session, Dr. Mukund. I have one question regarding, is there any possibility we can go with natural bioactive moieties with the self-emulsifying drug delivery system?
Yeah.
We
Yes,
have to
you
take
can go,
isolated
but
moieties
because
or we can take it as a whole
yeah,
extract?
yes. Very nice question. Actually, you can protect the API if it is for example proteinaceous in nature, or from the way you are asking. What I feel, that if you want to protect the API, you can protect very carefully by using this technique, and you can go ahead for the further formulation development of bioactive moieties also.
Okay. Thank you.
One more question is there, sir.
There is one question from Deepika. Shweta. Is there any maximum dose limit while selecting API for SADDS?
That skill that you have to develop based on the study, or decide based on the study, that how much amount or how much dose can be incorporated in the isotropic mixture. So for that you have to screen the maximum surfactant or surfactant which is showing the maximum incorporation, or oils which is showing maximum incorporation. As such, I found there is no limit of the dose that you can choose, but you can try to incorporate maximum dose. That will be the biggest achievement for you if you go for maximum dose API.
Okay. Thank you, sir. Indeed, it was a very informative session. You gave overview of self-emulsifying tablets with optimization. Few of our students are working on self-emulsifying drug delivery system. It helped them a lot. In future also, we will expect your help in case they need. We will contact you, sir. Thank you.
Yeah, definitely.
Thank you
It
once
will be
again,
my
sir.
pleasure.
Yeah.
Yeah,
Thank you once again for
thank you.
accepting our invitation. With this, we have come to an end of this session. We will meet again at afternoon session. Thank you, one and all.
Okay. Thank you, man.
Thank you, sir.